Hi, everyone, and welcome to episode 20 of the Cancer Researcher podcast, brought to you by the European Association for Cancer Research. In this episode, we will be discussing another interesting and impactful paper featured in the EACR's Highlights in Cancer Research, a paper that looks into the potential clinical benefit of activating oncogenic signaling. Cancer initiation and progression revolves around activated oncogenic signaling and suppressed expression of tumor suppressor genes. Oncogenes who are activated in cancer are responsible for inducing and fostering tumor development. Hence, common approaches in the attempt to fight cancer focused on the inhibition of oncogenic signaling, with various degrees of success. But a common culprit, cells eventually developed resistance to therapy. More recent research brings strong evidence that cancer homeostasis relies on a balance between activated oncogenic signaling and stress response pathways activated as a result of the inherent toxicity. Today, I have here with me Dr. Matheus Diaz, senior scientist in the laboratory of Rene Bernards at the Netherlands Cancer Institute, to elaborate on this and discuss a rather paradoxical proposal, as he calls it in his paper, to treat cancer by hyperactivating oncogenic signaling. I am your host, Dr. Alexandra Boitor, Scientific Officer at the EACR, and I invite you to delve into today's topic. Welcome, Matheus, to the Cancer Researcher podcast.
Thank you very much, Alexandra. It's a pleasure to be here.
It is great having you as a guest on our podcast, not lastly because this gives me the opportunity to meet Matheus, the researcher, in addition to Matheus, the chair of the Early Career Researcher Council, and discuss about your research for a change instead of brainstorming new ways of making the EACR community a supportive environment for early career researchers.
Yeah, that's actually true. It's a completely different setup that we are meeting this time.
I suspect it's going to be a rather different conversation than the ones that we've had in the past, but I'm really looking forward to it.
Me too.
Diving straight into today's topic, the paper Paradoxical Activation of Oncogenic Signaling as a Cancer Treatment Strategy, you recently published in Cancer Discovery together with your supervisor Rene Bernards and your collaborators. May I first ask, what triggered your interest to explore hyperactivation of oncogenic signaling as a potential anti-cancer approach?
That's a good question. And that question actually brings me back to the past. A few years before coming to the Netherlands, I was in the beginning of my postdoc working in a lab that was not really dealing with cancer, but it was investigating the cell cycle and how cells actually move through the cell cycle. And one of the tools that we were using back then to study the cell cycle was FGF2, which is a growth factor. So it was supposed to promote cell proliferation and promote progression through the cell cycle. But it turned out that the lab was using for a very long time a KRAS-driven cancer model. And in this KRAS-driven cancer model, FGF2, instead of promoting proliferation, it was stopping the cells in the cell cycle. For a very long time, the lab tried to figure out what was going on, why FGF2 was doing exactly the opposite of what it was supposed to do. The prevailing notion back then was that it was probably not based on the same signaling pathways that FGF normally activates, because FGF was not supposed to do that, but because I didn't have any background in cancer. So for me, it could be just that FGF was doing exactly what it was supposed to do. But because they were cancer cells, they were kind of on overdrive because of this activation. And that was exactly what I showed in this beginning of my previous postdoc. And that was what got me hooked on that idea that you could potentially overactivate oncogenic signaling. And with that, you would kind of overstress cancer cells and you could kill them based on those stresses.
Very interesting. This paradoxical approach, as you call it in your paper, is part of a new school of thought that started developing only in the past decade or so. Am I right?
Yes, it's hard to know exactly when it started to develop. As I said, in my career, it was something like 10, 12 years ago when I started studying that. But multiple lines of evidence popped up here and there. And if we really think straight, this notion should already be clear when we figured out that there was the very well-known phenomenon of oncogene-induced senescence. because it's not very different. It's just oncogenes or oncogenic signaling inducing a very strong stress response in cancer cells.
Throughout this study, you focused on inhibiting protein phosphatase 2A by using its inhibitor LB100. May I ask how or why did you decide to act on this phosphatase and not another?
The reason to use this specific drug is that when I started here at the lab of Rene, I was really interested in developing and expanding and maturing this concept of the hyperactivation. So I wanted to find things or activators that if we really understand how they work and if we really can come up with interesting combinations that we could test in patients. And it turned out that there are a lot of different inhibitors of oncogenic signaling, but not a lot of activators. So I didn't have many different options for that. And this drug, this LB100, this PP2A inhibitor, was already considered pretty safe in a phase 1 clinical trial as a single drug. So I knew that if I really find something interesting with this drug, I could potentially translate that into a clinical trial. And because PP2A is a phosphatase that inhibits many of those oncogenic signaling pathways that are normally deregulated in cancer cells, we figured that if we inhibit the inhibitor, we could get activation. And because the drug was already through a phase 1 clinical trial, we could potentially use this in patients if it works. So that was the only reason to choose exactly that specific drug.
So it was sort of a practical decision.
Exactly.
So what effects have you noticed following PP2A inhibition in your experiments?
Well, the first thing that we noticed was that indeed inhibiting PP2A was doing what we expected it to do, which was overactivating oncogenic signaling pathways in cancer cells, inducing a lot of stress responses and suppressing the proliferation of these cells. Despite the activation, it's not that they were cycling a little bit faster. It was exactly the opposite. They were getting stuck in the cell cycle, just like I observed very much in the past with the FGF2. They were like really getting stuck in the cell cycle because of this overdrive of oncogenic signaling pathway. So we observed these effects and then we kept moving on and investigating what would be the best way to target these overstressed cells.
So you decided to explore a dual approach, in a way, hyperactivate oncogenic signaling and control the stress response. You designed a custom stress-focused drug library of compounds targeting stress response pathways associated with malignant phenotypes, including some senolytic drugs, considering that senescence can be seen as a survival response of cells under stress. But your library, if I remember correctly, comprised about 164 selected compounds. So how did you decide on targeting V1 in the end?
Yeah, that's a good question because, as I said, when we started treating the cells with LB100, we noticed that a number of different stress response pathways were being engaged. So we didn't know which would be the best stress response pathway to actually target in combination with the activation. So that's why we designed this library, because with this library, we could inhibit multiple different proteins in these different stress response pathways that are normally associated with the malignant phenotype. And then we could just ask a very simple question. Which of those drugs would be more toxic in the presence of this activation? And then from this stress focus screen, we identified some check-1 inhibitors and also WEE1 inhibitors. These inhibitors became more toxic in the presence of the activation. And because we had also a parallel CRISPR screen that, again, identified the knockout of WEE1 as being synthetic lethal with the inhibition of PP2A, we decided to focus on this combination of PP2A inhibition plus WEE1 inhibition. And also because the clinical development of the WEE1 inhibitors by the time that we identified the combinations were a bit further compared to the development of the CHEK1 inhibitors. So it was a pretty clinically relevant combination.
So now that I have a better understanding of the concept and drugs of choice, how does it all work? What are the effects that you've noticed on cancer cells?
Yeah, the thing is, when we went to identify the mechanism, one thing that was the starting point was the mitosis, because in the end, PP2A itself has some effect on the mitosis, but also WEE1 is best known for being a gatekeeper kinase controlling mitotic entry. So if cells have any problem during DNA replication or DNA damage or things like that, WEE1 tends to block the cell cycle and then to prevent these cells to enter mitosis. So if you inhibit WEE1, they enter mitosis with DNA damage, which is definitely bad for cancer cells. So first thing that we actually look was to look at the mitosis, and what we observed immediately was that the combination of the two drugs, each one had some effect on the mitosis, but the combination of the two drugs actually led to pretty much all the cells, when we were using this time-lapse microscopy, all the cells end up stuck in mitosis, in a very aberrant mitosis, up to 24 hours. And we also noticed that if you give them a little bit more time, they would start dying slowly in the presence of the combination, while they would recover to their normal cell cycle after a few hours if you just treat with the single drugs. And then we even took a step back to figure out what was going on during the DNA replication. And what we noticed was that these cells, in the presence of the combination, the DNA replication was literally collapsing. So you had a collapse of the DNA replication, and these cells were moving from the S phase straight to mitosis without even finishing DNA replication. And that was obviously very toxic to these cells. So that was more or less how we identify what was the phenotype, what was going on, why the cells were dying in the presence of this combination.
Another thing that I wanted to ask you is that, well, both adavosertib and LB100 have been proposed as sensitizers for chemotherapy in the past. Adavosertib in preclinical studies, and LB100, I think it even reached clinical investigations. So how does the combination of LB100 and adavosertib compare with each of these drugs in combination with chemotherapeutic agent?
Yeah, that was in the end a very important question, because when we identified the combination, we also noticed that both drugs were being tried in the clinic in combination with chemotherapy. That was a very important and obvious experiment that we had to do. And we actually tested side by side the combination of adavosertib and two different chemotherapy drugs, and the combination of LB100 plus two different chemotherapy drugs, and the combination, obviously, of LB100 plus adavosertib. And the combination of LB100 plus adavosertib was superior of all the other possible combinations. So it was pretty clear that you had much more synergy when you combine LB100 and adavosertib than when you combine each of the two drugs with the chemotherapy drugs.
Moving on a bit from how these drug combinations work and getting back to the effects that they have on cancer progression, I'd like to touch a bit on the subject of resistance to therapy. Most, if not all, anti-cancer therapies proposed have over time determined tumors to develop mechanisms to resist or escape therapy. From your paper, I understand that the approach you are proposing is no exception. However, you observed a very interesting, and I'd dare to say not at all detrimental, effect of therapy resistance in this case. Would you like to tell me a bit more about how you studied the acquired resistance to the combination of LB100 and adavosertib and its tumor suppressive effects that you observed?
Definitely, because that's also very interesting, because in this manuscript, we decided to really go from hypothesis to identify the combinations to mechanism and to the tests in vivo. But also, it will be very important to address resistance, because in the end, it is the major challenge for cancer therapy. But if I may, it's important to make a small and even logical inference. We are very familiar with the scenario that we have for resistance to either chemotherapy or even targeted therapies. Because in the end, you inhibit the cell proliferation or the signaling pathways, and then you have some effect. And then after this effect, the cancer cells manage to rewire these signaling pathways. And when they do that, they tend to become even more malignant than they were before, and even more difficult to treat with the next drug. But that happens when you are inhibiting oncogenic signaling. So we envision that because our treatment was actually overactivating oncogenic signaling, it would be just logical to expect the opposite, because to really survive this kind of treatment, cancer cells would have to either turn down some of their oncogenic signaling pathways or reactivate some tumor suppressor genes. So that was more or less what we were expecting. And we were quite fortunate because from the very beginning of this investigation, even in CRISPR screens, we were already observing that if you knock out some oncogenes, the cells would become less sensitive to this overactivation. By the same token, if you overexpress some oncogenes, the cells would become more sensitive to this overactivation. But the real proof was that we decided to select cells in vitro that were resistant to this overactivation of oncogenic signal, to this combination that we were proposing. And then we observed a number of different interesting things, just to mention two of them, that these cells were actually now suppressing oncogenic signaling, even in the presence of the drug, which was exactly what we were expecting. But we even noticed some more dramatic changes because one of these models that we tested was highly aneuploid. It had something like an average chromosome number of 70 or so. And then after becoming resistant to the combination, they have on average a nearly diploid karyotype. So they become much less aneuploid than they were before. But the real proof was to inject these cells now in mice. And when we did this, we noticed that these cells that became resistant to the combination, they were now either failing to grow tumors in mice or they were growing much smaller tumors, indicating that after becoming resistant to this approach, they became less malignant, what we called tumor suppressive acquired resistance. So it was really interesting to notice that the prediction that the resistance should go on the opposite direction actually materialized on the experiments.
That sounds great. So basically, either the treatment works and kills the cancer cells, or the cancer cells become resistant but lose their tumorigenic potential.
That's the idea. Let's hope it also works like that in patients, right?
Sounds like a win-win to me. How about the toxicity of the proposed treatment approach? Did you notice and or can you foresee any side effects?
Yeah, that's a good question, a very difficult one to answer because obviously during the manuscript, we performed tests in non-transformed cells in vitro, and we also observed body weight in the mice and even analyzed a few vital tissues on these mice that were treated by the combination. But this is all experimental work. The real proof that the combination is not toxic, we can only see actually when we have clinical trials, because in the end, we are very familiar with the situation that a number of drugs or drug combinations work very well in animal models, but they don't work at all, or they are highly toxic for patients. So in this particular case, we are very aware that the clinical trials should be performed very carefully because we really need to be aware of potential toxicities arising for these combinations, especially because we didn't see much context dependence in terms of what are the cancer cells that actually respond. Because most of the cells that we tested on the manuscript actually responded. So we really need to be careful when testing this in the clinical setting.
So what I noticed in your paper is that your experiments extended over the period of 1 month. I would expect that in clinical practice, that probably wouldn't be enough. So do you have any plans for additional long-term in vivo experiments before you move into clinical trials?
Yes, we are actually conducting a few long-term experiments right now. I'm not entirely sure if they help very much to predict what's going to happen in patients, because as I said, we are now all too familiar with the situation that things work perfectly in animal models, but don't work at all in patients. So I think it's an important control, but the real deal is actually to test in patients, being very careful with the doses and with the monitoring of these patients.
So you show in your paper, as I think you've already mentioned, that this drug combination hyperactivates multiple oncogenic signaling pathways and simultaneously engages several stress response pathways. Is there a risk that this approach could induce tumorigenesis in normal or precancerous cells?
Well, that's the question that you get in all presentations, right? When you finish presenting, the first hand to ask the question, that's exactly the question that comes.
At least it wasn't my first question, but it's still on the list though.
No, definitely, definitely. This is an important question. There are a number of different sides to approach that question. I tend to prefer to approach first from the more conceptual perspective. Because if there is one thing that we know about cancer cells is that they have increased oncogenic signaling, detect feedback to control that signaling, and tonic stresses. So because of these intrinsic characteristics of cancer cells, we should expect that they are much more sensitive to this approach compared to normal cells. So there should be a therapeutic window, particularly because normal cells, by definition, they must have some intact feedbacks to control oncogenic signaling, because if they didn't have this feedback, they would be cancer cells and not normal cells in the first place. So they should have those feedback to control oncogenic signaling. But there's also a more pragmatic approach, because we have, for instance, not LB100%, but we have the drug lithium chloride that has been used in clinical practice for bipolar disorder for many, many decades. And despite the fact that the lithium chloride is a GSK3 beta inhibitor and therefore activates the Wnt pathway, which is a major driver for colorectal cancer, when they looked at these patients that were treated with lithium chloride, they could not observe any increase in cancer incidence. If anything, there was a slightly decrease in the incidence of gastric cancers in these patients that were treated for decades with an activator of oncogenic signaling pathway. So it looks like that pre-malignant cells or even cancer cells, they are not waiting for more signaling to grow. They're actually waiting for something else, maybe for aging or for inflammation, but not necessarily more signaling. More signaling seems to be harmful even for pre-malignant cells.
It really seems like a great approach, at least on paper from preclinical studies. So my last question would have been, what are the next steps for this study? But you've already mentioned you plan on taking it into clinical trials. So I'll rephrase my question and ask you if you have any sort of timeline in mind for the beginning of the clinical trials, and if you're planning on further investigating the molecular mechanism into more detail in parallel to that.
Yeah, these are very important questions. I used to give a timeline for the clinical trial. I used to say, oh, we're really expecting to have the clinical trial in the next coming month. But I've been doing this for quite some months already. And I realized that the amount of bureaucracy that are involved in those clinical trials, it is really immense. And there are a number of unforeseen events that you cannot really control and that are totally outside of the lab. They are like legal aspects of clinical trials. So the real answer is that I really don't know when the clinical trial is going to start. We have the protocol written, we have agreements with companies and with the hospital, with our phase 1 Unity, but it really depends on a number of bureaucratic details that are completely outside of my scope. And for the further investigation, it's important to mention that this combination is only one combination. The approach, the idea that you can essentially overactivate oncogenic signaling in cancer cells and combine that with stress targeted drugs, it's a concept that is much broader than this simple combination. So what we are doing right now is try to identify other combinations based on the same concept and try to understand for each of those different activators or different drug combinations, understand exactly what is going on and how we can make it better and how we can develop new activators, given that we, as I said, we have a number of different inhibitors, but not a lot of activators. So the idea is to exploit this concept now in a much broader way for different cancer types, different drugs. So this is really a completely new and very much untapped field of research, which is much bigger than the combination that we actually propose right now.
Well, everything we discussed today points towards a very promising approach on the horizon for the treatment of cancer. The data presented in your paper and discussed today shows considerable context independence of the synthetic lethality between the two drugs used, namely LB100 and adavosertib. As you used models from diverse genetic backgrounds, you tested 7 different colorectal cancer cell lines with various driver mutations and different tissues between the in vitro and in vivo experiments. I think you've looked at colon cancer, pancreatic cancer, and bile duct cancer. So thank you very much for having this conversation with us and presenting your research on the Cancer Researcher podcast. Thank you for allowing us a glimpse into your research by accepting our invitation.
Thank you very much. It was really a pleasure to have that conversation with you.
Thank you. We really hope that you enjoyed listening to this fantastic research story and that you learned something new from us today. If you have any follow-up questions regarding this conversation, please don't hesitate to contact Matheus by email. Please subscribe to our podcast so you won't miss an episode. You can find The Cancer Researcher on your podcast platform of choice. Subscribe and tell your friends about it. And don't forget that you can also find us on Blue Sky and Instagram at Hello EACR, and you can search for us on Facebook and LinkedIn. We welcome feedback and suggestions for other topics or preferred guests through our feedback form, which you can find on our website at www.eacr.org/podcast. Talk to you soon.